Good morning, good afternoon, good evening, and even good night for some of you. And welcome to our webinar series, New Tools of the Trade in Life Sciences, Technology that Enables Breakthroughs. Today's webinar will focus on laser microirradiation and real-time recruitment assays using an engineered biosensor of nucleosome ubiquitination. My name is Renate Weitzbauer. I'm the lead editor of this webinar series here at Bioprotocol. For those of you who are not familiar with Bioprotocol, it's an open access online journal whose mission is to make research more transparent and reproducible. Before we begin today's webinar, I would like to make a few quick announcements that might help you to get the most out of this webinar. If you have questions, please feel free to post them in the Q&A or in the chat section throughout the talk, and Carolina will address them after the talk. In addition, as Antoine already mentioned, the webinar will be recorded and posted on our website later. So if you want to revisit it, you can do so at a later time. It is my distinct pleasure to introduce the moderator of today's webinar, Antoine de Moret. Antoine is currently an assistant professor at the Department of Biomedicine at Aarhus University in Denmark. He received a PhD from Leiden University in the Netherlands, a postdoctoral certificate from Stanford University, and an Ignite certificate from Stanford University Graduate School of Business. Antoine's lab investigates the mechanisms that enable adult stem cells to regenerate tissues, with a particular interest in stem cell quiescence, its function and regulation, and his work has been published in high-profile journals, including Nature and Science. For his research and leadership, he has received several international awards, including the highly competitive development grant by the Muscular Dystrophy Association for his work on preventing the production of the toxic protein DUX4. In addition to his scientific career, he also regularly teaches communication and resilience to science trainees, including extensive workshop, and has co-wrote the book Magnetic: How Great Leaders Persuade and Inspire. And with that, Antoine, thank you so much for being here today, and the floor is all yours.
Well, thank you, Renata, for that wonderful introduction, and thank you all for tuning in. DNA damage response pathways are central to the survival and function of every cell. These pathways, they depend on the tight regulation of protein localization and levels. However, protein modifications that regulate this are often difficult to study in live cells. Now, today, we're going to hear about methods using avidity-based genetically encoded sensors for one such protein modification, namely histone ubiquitination. With us is Dr. Carolina dos Santos Passos, a research scientist at the Department of Biochemistry and Molecular Biology at Colorado State University. Carolina trained in the field of protein biochemistry during her PhD at the Federal University of Rio Grande do Sul, in Porto Alegre, Brazil. She identified numerous chemicals isolated from plants that could affect acetylcholinesterase activity, which could impact the progression of Alzheimer's disease in models. Then she moved to the University of Geneva, where she worked on understanding the regulation of deacetylases implicated in the progression of neurodegenerative diseases. And this sparked an interest in ubiquitination. And she is now working on genetically encoded ubiquitin sensors, and most recently contributed to a study revealing how neurotransmitter synthesis enables synapse formation. Carolina, thank you for joining us today. We look forward to your talk, and the floor is all yours.
Thank you for the introduction. And I think I need to share my screen. But so this talk is divided in two parts. First, I will talk about our design sensors and the strategies that we use to develop these tools, and also how they are characterized in vitro and in mammalian cells, and some of the applications that we are trying to pursue. In the second part of the talk, I will talk about a protocol that was published by Bioprotocol recently. That's the laser microirradiation and the real-time recruitment of our header of histone ubiquitination. So ubiquitination, it's the covalent attachment of ubiquitin, that's a small protein, small globular protein containing 76 amino acids, to lysine residues in substrates. And depending on the number of lysines that are modified, we can have monoubiquitination with only one lysine, but we can also have substrates that have multiple lysines that are ubiquitinated, resulting in multi-monoubiquitination. And in addition, ubiquitin itself, it has 7 lysines that can be ubiquitinated, and this can result in a large variety of polyubiquitin chains. There are 2 main functions of ubiquitination in cells. The most studied, its most well-known function, it's a degradative function that its proteins are ubiquitinated and then target to proteasomal degradation. But the main focus of our research, it's ubiquitination as a non-degradative function, as having a signaling function. And here we have histone ubiquitination in nucleosomes. Histones are the most abundant ubiquitinated substrates. And in nucleosomes, there are 4 main sites of ubiquitination that are the most studied. And these are the N-terminal and the C-terminal tail of histone A2A, and also the C-terminal tail of histone A2B. And depending on the lysine residue and the histone that are modified, ubiquitination, you have different functions. And ubiquitination, so it's a histone post-translational modification, such as phosphorylation, methylation, acetylation. And this ubiquitination of histones A2A and A2B, it's highly specific, and it's mediated by writers, proteins or writer enzymes that in this case are ubiquitin E3 ligases. The erasers of this PTM will be deubiquitinating enzymes, and the readers will be molecules or effectors that will be recruited by these different types of histone ubiquitination. Here, our main subject of this talk and the sensor that we developed aim to identify H2A, K13 and K15 ubiquitination, that both of these lysines can be modified, and they have a function in the DNA damage response. The E3 ligase that mediate this PTM, it's RNF168, and among the identified writers, we are mainly interested, or the most important for us, are 53BP1 and the heterodimer BRCA1 and BARD1. So what are the tools that you have to study nucleosome ubiquitination? These modifications can be addressed by using or identified by using antibodies, or also mass spectrometry approach. But we also can have antibody substitutes, such as single chain fragment variable antibodies and nanobodies, and also non-antibodies alternatives. And among these, we have our designer readers. And to understand the concept behind our designer headers, first we can have a general understanding on how headers of nucleosome ubiquitination or of histone post-translational modification works. So we have the nucleosome, that's our substrate, and we have ubiquitin that will be covalently attached to this nucleosome to an isopeptide bond. So the headers, they are composed mostly of 2 binding domains. One binding domain that we call the anchor, that will bind to a surface in the substrate, that's in this case, it's the nucleosome. And a second bind domain that's a ubiquitin-binding domain that you bind ubiquitin. When these two independent motifs are connected through a linker, we'll have a decrease in the entropic cost of binding, and also this will determine the specificity to bind one form of ubiquitinated nucleosomes over the others. So take into account this strategy, or like this mechanism, we then used this avidity-based design, and we developed a strategy to generate these sensors for ubiquitinated nucleosomes. So for doing this, we need to have structural models that can be available from crystal structures or cryo-EM structures that we have in PDB, or you can also build these models. So next step, we want to identify potential anchors and ubiquitin-binding domains that would be used for these sensors. And to test then in vitro, to test the binding, you need to obtain nucleosomes that have the modifications that you are interested in studying. Then we can test the prototypes for evaluating its specificity and also binding affinities. And having this data, sometimes we need to optimize linkers by structural models, or a strategy that has been shown very useful to us. It's molecular dynamic simulations, because it takes into account all the conformations that ubiquitin can adopt when attached to the nucleosomes. And then we can test the sensor in vivo and apply for detecting these modifications. So to generate our prototypes, that first we designed two sensors that target either A2AK15 ubiquitination or A2BK120 ubiquitination. And so we select anchor motifs that were peptides that bind to a acidic surface in the nucleosome called the acidic patch that it's shown here. And these are two viral peptides that are structurally resolved, LANA and I1. And we select also ubiquitin-binding domains that are the UBDs. And for this, we started with TSR, that it was a binding domain that was genetically, that was engineered in our lab, and the UBA domain of ubiquilin-1. These independent motifs, so when they bind with low to moderate affinities to the individual targets, and we then use linkers that can be rigid linkers, such as an coiled alpha helix, or flexible linkers, to then generate this protein that it's designed to bind to ubiquitinated nucleosomes with affinities. Ideally, we want affinities that are in the range between 5 and 100 nanomolar, and these are ideal to detect what we want to detect in cells without or with minimal disturbance of the signaling mediated by effectors of this post-translational modification. So with this strategy, we generated HIDER1, and to HIDER1, we took advantage of LANA and how it binds to the nucleosome acidic patch, in which we have the N and C terminal tail that are faced to this surface where you have A2A-K15. And we used a rigid linker, a coiled coil alpha helix, to connect LANA to the ubiquitin-binding domain. And so we generated the HIDER1, and with the goal, just to remember, to detect this A2A-K15 ubiquitination that's generated in response to DNA damage. So to characterize this sensor in vitro, we expressed the protein in E. coli and purified from E. coli. And we used maleimide chemistry to link or to attach a fluorophore molecule to HIDR1. And when we titrated this fluorescently labeled HIDR1 with ubiquitinated nucleosomes at increased concentrations, we observed that this fluorescence also it's increasing the fluorescence of the HIDR, and this allows us to determine the binding affinities of the sensor. So we have a panel of different ubiquitinated nucleosomes, and when we tested the binding of HIDR1 to H2AK13 and H2AK15, we saw that the sensor is binding these nucleosomes with an affinity in the range affinity around 5 to 10 nanomolar. And so these affinities are suitable for the applications in live cells. And looking then for some of our in vivo characterization, we observed that in mammalian cells, HIDR1 doesn't significantly bind to other forms of ubiquitinated nucleosomes that are not K13 and K15UB. I'm not going to these details because of time, but if people have questions, we can discuss this later. So now the question is whether HIDR1 can detect this ubiquitination induced by DNA damage in cells, in mammalian cells. So to characterize HIDR1, or like to characterizing that the sensor can detect this modification in cells, we performed the live cell measurements in which we genetically encode HIDR1 and expressed the protein the sensor fused to GFP. And we use the UV405 laser in the microscope to induce damage in a strip in the nucleus. And we observed that 15 minutes after laser microirradiation, HIDR1, it accumulates to these laser tracks. So it's showing that it's being recruited to DNA damage sites. In addition, we can look here that there are some small foci in cells that are unperturbed. And here we have other examples. So besides of detecting or being recruited or accumulate in DNA damage induced by laser or IR, we also have this nuclear foci suggesting that our HIDR is also accumulate at endogenous DNA damage regions in these UTOS cells. And these both the recruitment of HIDR to DNA double-strand breaks induced by microirradiation, and also this focal accumulation in unperturbed cells, it's dependent on the RNF168, that's the E3 ligase responsible for adding this post-translational modification, and this suggests that our HIDR it's detecting H2AK13 and 15 ubiquitination at the DNA damage sites. So now that, okay, so we are expressing this protein or expressing this sensor in cells. And it's important that we have levels that are tight controlled, because our HIDR will recognize this post-translational modification, but we don't want that it competes with effectors of DNA repair. One experiment that we did to demonstrate or to try to see what are the levels of expression that we can work at, we look at if the header affects the repair, the kinetics of repair of DNA damage induced by gamma irradiation. And we compare the kinetics of repair measured by the appearance and disappearance of gamma H2AX, that's a marker of damage, in cells not expressing header 1, as you can see here, and in cells expressing header 1 at the levels that we worked in the previous experiment. And if you look at the kinetics, we have a maximal accumulation of gamma H2AX 2 hours after damage, and this goes to levels similar to the non-irradiated cells after 24 hours, and the curves for the cells that don't express header 1 and that express header 1 at the levels that we use in our experiments were similar. So now that we have this reader, what can be it used for? So one of the applications, or the main application when we started working with these sensors, was for live cell imaging. So to see in real time how this modification, how dynamic are these post-translational modifications. So one of the experiments that we did, we took advantage that we can observe header 1 accumulated, So we took advantage that header 1 accumulate in foci, and we use cells that express this PIP future reporter, and the differential expression of these two proteins allows us to determine where the cells are in the cell cycle. And by monitoring the header 1 or these nuclear foci, we can see how H2AK13 and 15, they are regulated, or how they appear and disappear throughout the cell cycle. And here I'm talking about the protocol, or the method that was published in Bioprotocol to measure the recruitment of header to DNA damage induced by laser microirradiation. So for these experiments, we used the U2S cells that we generated in our lab, and that they stably express header 1 fused to GFP and this expression, it's controlled, or it's under the control of doxycycline. So we plated the cells in dishes suitable for live cell measurements. And we, after the cells attached, we transfected them with M-cherry 53BP1 FFR. That's the minimal, the focus formation region of 53BP1 FFR. And this, after 16 hours, we add doxycycline to induce the expression of header 1. And we can observe header 1 accumulating or present in the nucleus about 4 hours after transfection. And ideally, with this concentration of doxycycline, we want to make the measurements between 4 and 12 hours after the transfection. So we have cells that are co-expressing these two headers of H2AK13, 15, and then we do laser microirradiation. So for the settings, or how to set up our system, we use the Zeiss LSM 880 microscope, but this can be adapted, or these experiments can be performed and any confocal microscope that has similar settings and equipped with 488 nanometers laser and 561 nanometers laser. So first we use open the software and connect the system. And we needed to set up the temperature, since we are working in live cells, to 37 degrees, and the CO2 atmosphere, that for U2S cells, that's the cells we are working, that's 5%. Here in number 2, we need to select the lasers that we are going to use. So we have GFP, we are using the argon laser operating at 488 nanometers. And we have the 561 laser that's also needed to turn on. So sorry that it was too small. So then we needed to select the conditions or making the setup to imaging these 2 colors, that's these 2 proteins. That's either one that's fused to GFP, and 53BP1 FFR that's fused to NHEJ. So we needed to select the tracks and which laser we are going to use to excite the sample, the pinhole size, and the master gain. Going to number 2, we use visible light that will be for NHEJ 561 nanometers and GFP 488 nanometers. So we need to select the mean beam splitter, and here you can use the same for both lasers. And considering that we use the UV light or the 405 nanometers laser to induce damage, we also need to select the mean beam splitter that will reflect the 405 nanometers laser. Then we need to select with which objective we are using. In these experiments, I use that 63x objective. And after selecting the objective to focus the cells and selecting the scan area, that we can zoom in the cells that we are going to irradiate. And this is important to do this first before setting up the frame size in optimal and the pixel dwell or the speed to maximum speed. So this is similar, these settings, these setups are similar for both fluorophores that were working NHEJ and GFP. And finally, we need to select which detector we use for each of the fluorophores and how much of the emitted light will be processed. And this will depend on the fluorophore that we have in the sample. So then we needed to set up the settings for laser irradiation and for the real-time measurements. So in the ZenBlack software, we needed first to check the options, time series, bleaching, and regions, and open the respective windows. In time series, we need to select the number of cycles, that's the number of frames that we acquire, and the time interval. Here we have 50 cycles that will be acquired in intervals of 20 seconds, and this you count for around 15 minutes of total time. For the bleaching settings, we need to see how many scans we want to collect after inducing damage by laser microirradiation. If you want a different scan speed for microirradiation, and this is ideal because we have more time that this laser will be exciting the sample and we have a higher extent of damage. And finally, to select which laser source we are going to use and the laser power. And these are all settings that needed to be tested because it can vary from sample to sample and from setups to setup. So it depends on the laser and the sources that you have in your microscope. And finally, we needed to select the regions that we are going to bleach. And in this system, we select so a strip in the nucleus. And here it's important that the height of this strip it's constant, or like we want to have the same right in all the experiments. And here we are setting in 5 pixels. And this will be dependent on the frame size that we set up before when we were looking at the individual channels. So we have our output, or our one example of one experiment. So this is the real-time recruitment, in which we have the curves of mean fluorescence intensity for 53BP1 FFR that is labeled to mCherry, and the other one that's labeled to GFP. And we see that in this region of interest that was drawn according to the size or the accumulation, we see an increase in the mean fluorescence intensity in both the HIDR and 53BP1 FFR. And in addition, we add a second region of interest that here is shown as ROI2, yes, 2, and this will be used for background subtraction. So having these results, we can calculate, or you can estimate the kinetics of recruitment of both of these 2 readers by first, so we can export these tables, and we can can calculate the normalized intensity according to what's shown in the box. And we can see that when we calculate this normalized intensity, our reader and the endogenous reader, or the reader that's previously used to detect this post-translational modification, they accumulate and are recruited to laser-induced double strand breaks with similar kinetics. So in summary, or the summary of this talk, the HIDR one, it's a avidity-based sensor that's selective to detect A2A, K13, and K15 ubiquitination in vivo. And we show that it can be recruited to double-strand breaks, and it's dependent on the heteroligase activity of RNF 168. And it can detect this post-translational modification in both cells that were subjected to damage, but also in cells that are unperturbed, that what we call endogenous damage. And we need when we express these readers as in genetically encoded in mammalian cells, the levels need to be controlled to minimize the competition with the endogenous DNA repair machinery, and to not affect the kinetics of repair. And the reader, it's recruited to DNA double-strand breaks with a kinetics that's similar to a red characterized endogenous reader. And it also can compete with endogenous effectors of the signal that are 53BP1 and the heterodimer BRCA1 and BARD1. So as future directions of this big HIDR project, we are currently working in sensors that target other A2A modifications, so K118, 119, and 127 ubiquitination, and in the future we also want to look at H3 ubiquitination. And this strategy, we anticipate that can be designed or can be used to design HIDRs for other histone post-translational modifications, such as methylation and acetylation. So I'd like to thank some people working in Quen and Yao Labs, and also our collaborator, Chris Snow, and especially Jung, who is the person behind the design of HIDR. So thank you for your attention, and I'll be happy to reply or answer to your questions.
Thank you, Carolina, for a wonderful, wonderful talk. Two exciting parts, one more generalized and then a more detailed application. We have a lot of questions coming in, but I would like to start with just some general questions overall. You mentioned at the beginning that histones are the most ubiquitinated protein species.
Mm-hmm.
Would you explain a little bit more what you mean by that? In what way are they most ubiquitinated?
So there are these different histones that are ubiquitinated, and there is a histone HA lysine 118, 119, and these around the, depends on the paper and the cell lines, but around 10% of these histone HA is ubiquitinated at 118, lysines 118 to 119. And these, when experiments are performed, where the pool are performed, a lot of ubiquitin that's detected, like ubiquitinate substrates, correspond to this histone HA. So okay. It's for ubiquitination, it's in comparison with other proteins, they are the most abundant, and also cells that have a lot of stones, and all the cells have, so I think it makes sense. But the bulk of histone ubiquitination, it's this histone HA K1 lysine 118, 119, and this it's present in regions of chromatin silencing, so it's responsible for silence maintenance.
Okay, thank you for clarifying that. So it's a large fraction of the histone 2A,
yeah,
and that
a large
itself
fraction.
is
Some papers they report that 10% of the total of histone HA is ubiquitinated, like normally without any process, just the maintenance of silencing.
Yeah, great. Then at the beginning, you also mentioned the different types of linkages that can be formed between ubiquitins. Do you know if it's possible that different types of links occur within the same ubiquitin chain? And could that in any way affect the sensors you develop, or is that distance always going to be too big?
So you mean okay, for some of them, we have mono-ubiquitination. For example, we can have H2AK19 that's mainly mono-ubiquitinated. If we have H2AK15, it can have polyubiquitin chains. And these are believed to be K63, so a type of linkage. It may affect the recruitment of headers. So for what has been published, the 53BP1 can bind or can still bind if there is polyubiquitin chains. But BRCA1, BARD1, it's only recruited by nucleosomes that are monoubiquitinated at this position. Don't know if I answered your question.
Yeah, no, that's exactly the kind of clarification. So there is a lot of knowledge already on the type of ubiquitin species that are recruited here, because that's I think pretty important in designing the sensor.
Some headers have been characterized that the headers that we developed, they were developed to also recognize if there are polyubiquitination, but we haven't tested it. So it's something that would need to be tested. Yeah,
I imagine that for that to work, you would need a longer linker.
Yes, there is another publication, not sure if it's already, but from Bob's lab with Jung, that they worked in a sensor for PCNA, and the PCNA can be ubiquitinated by K63, and they have a longer linker, and they have also tandem ubiquitin-binding domains that will recognize the K63 chains. I think it was under review, so I don't know if...
It's something to look forward to. In terms of designing the linkers, are there any properties to keep in mind when making a sensor like this?
One of the, okay. So we can use rigidity, and this we can direct, or we can constrain the mobility of the sensor, and this will decrease the entropic, or the entropy of binding. And you can also have selective for this. Another constraint, or another thing that you need to keep in mind, it's the distance or the length of this peptide that you are using as linker. In these sensors, HIDER 1 and HIDER 2 that are our prototypes, we have the anchor that will bind the acidic patch, and we have a minimal distance that around 30-40 angstroms for A2AK15 ubiquitination. For A2B-K120, I didn't present the results here, but we did the molecular dynamics, and for molecular dynamics, we saw that the conformations that are like better are the conformations that we can distinguish these two different ubiquitination sites. They have a shorter distance between the anchor and the ubiquitin-binding domain. So we opted for not using any linker. So we directly connect, in this case, IU1 to UBA, and the HIDER show to be selective and to detect A2B-K120. So generally, I would say the rigidity of the linker that will be better to give specificity, and also to reduce the entropy of binding, and also the length that it will depend on the position where ubiquitin is attached.
Thank you for clarifying that. So it sounds like it's going to be really important that you have some good structural insights before you design. What tools do you recommend to make or analyze the structural models?
So most of them, it's kind of guessing for now. We use, ideally, we take models that are available in the protein data bank. So for LANA and IU1, we choose these anchors because we have structures of these anchors that were published in PDB. So we knew how they fold and how they bind. For tools, we are using GROMACS and for molecular dynamic simulations. And our collaborator, Chris Snow, we have projects now in collaboration with him, in which he's using AlphaFold generated models, so to see the conformations and try to optimize the headers. And the main goal is to optimize the linkers. So now we are pretty excited because he generated, or he made like some structures, and we are going to test them.
So the idea is to use AlphaFold to basically predict structure when there is none, or use a database when there is actual structural information, and then somehow dock the two and see how they fit and work from there.
Yes, this we did some docking. So when we, more when we have the molecular dynamic simulations, that we have all the conformations that we sampled for these two modifications, then we dock the anchors, but we dock the ubiquitin-binding domains. So we have those conformations, and we dock the ubiquitin-binding domains, and we knew that for A2B-K120, UBA have like a preference, or could dock to most of the conformations over the TSR, that's the other UBD. What we can't predict as much with that approach was the linker, and I think this AlphaFold may help us.
Yeah, I can imagine that
Yes, if you want a rigid
flexible.
linker and if you have a helix, this it was difficult with our initial approach, but not only AlphaFold, but some publications, some scripts that were published recent that we can have some ways to predict the conformation of the linkers.
Great, thank you for clarifying that. I have some questions here on when you take your model into the lab. What's the number of permutations in the linker in the binding chromates that you had to test before you ended up with a functional sensor? Or did everything work right away?
That one worked right away.
Okay, fantastic.
Okay, it was kind of lucky because we tested for different nucleosomes in vitro that I didn't show the results. And in vitro, it also bound to A2AK120 ubiquitination. When we tested in cells, it was the binding to A2AK120, A2BK120 and A2A119. It was like almost like the anchor alone, and when we don't have ubiquitination, so this was kind of lucky. And it's something that we didn't consider because in our in vitro experiments we use isopeptide mimetic bonds, so we do chemical cross-linking, and these have properties that are not exactly the same as native bonds. So for this, either one worked fine. We needed to do the mutations, for example, for fixation, or we did some mutations to have a binding affinity that was slightly decreased. We are testing other headers now for those other forms of ubiquitination, and these we are testing. We start testing 10 of each.
Okay, that's good to hear that sometimes there is luck in science.
Yes, yes, but yeah, for this okay, this one that's K127 ubiquitination, it's more complicated because it's in the C-terminal tail, so it's very flexible, and this we are testing more versions of the reader.
If you are looking to do lots of permutations, you mentioned quite a range in KDs that are optimal, mostly nanomolar range. But what would you say is a minimum in terms of KD value to be workable?
If it's higher, or if higher than 100 nanomolar, we start to lose. We don't have the resolution for this. Ideally, between 110 and 100. There is some of the sensors that are 200, 250 nanomolar that we are testing now, but not in the micromolar range because it will only detect when the levels are too high, and we start to lose, we don't have a resolution, or we can't measure, especially when it's the endogenous signal. And can I add something here?
Of course.
Yes, it also depends on how much we have of this modification in the cells. So if it's A2AK19 that we have ubiquitination that we have like 10% that's modified, we can work with like higher KDs. But if it's something that's very low levels, we need higher affinity, so lower KD.
Yeah. That makes perfect sense. I mean, related to that, and follows up an application question, whether you've been using stable lines mostly, but could you use transient expression, and would expression differences across cells impact the actual reader function?
Yes. So transient, yeah. So
that's clear.
Yes, we opted for stable expression. So doxycycline control is better because we can use low concentrations of doxycycline when we have low levels. When it's transient expression, we will have a range of expression levels, and when the sensor to express at very high levels, this will affect the downstream signaling, and the cells will stop in divide and can be toxic to the cells. So we can use transient transfection. We can use promoters that, like not CMV promoters that are low expression, but for our experiments, we are opting most for stable expression.
If we follow up on that nice track of the 53BP1, I have one question here: how that region is selected in the microscopy software?
So the region for bleaching or for irradiation, it's a rectangle. For selecting the region, we have the options for this software for analysis. We can use the rectangle, or you can use this closed polyline that we can add the region of interest that correspond to where we have the enrichment of the sensor.
So, and then when you set this up, do you take into account where there might be nucleoli, or does it matter where in the nucleus you select your region? Sorry,
I didn't understand the first.
Does it matter where in the nucleus you select your region of interest? For instance, you do nucleoli.
When for the irradiation, it shouldn't matter. I select kind of the experiments kind of vary, so we have here, but it shouldn't matter.
Great, and I have a couple of questions that delve into the applications of the sensor. Basically, can reader 1 read any ubiquitin site on a nucleosome, or could it be adapted to do that?
Sorry, other sites, they mean? Not exactly. We have we have a version that's has a flexible linker that's 18 amino acids that we call pen reader, and these we show that can bind to all those modifications that I show in the beginning. But these we have so differently of either one, that we have a coiled coil that's rigid. We have a 18 amino acids linker, and it's serine, mainly serine glycine, so it's a flexible linker. And these we use as a pen nucleosome reader. So I don't know if I replied the question, but this we can read any of the modifications, and the KDs are depending on the nucleosome are like 10, 20, and maybe 100 nanomolar.
Yeah, along the same lines, you could wonder if the sensor could potentially be adapted to target epigenetic modifications, or maybe even to be developed into a therapeutic, right? That you link to things that could modify the process.
This I don't know exactly. Maybe
very far out.
Yes. A lot of headers are they use these acidic patch, so they have these anchors, and we anticipate that we can attach bind domains, or can use bind domains and target different modifications. So this it's true. Our goal is more for detection, to show this and understand how what's the roles and what's the importance of this modification for medical applications. I don't know how we can certainly use in animal models. It's something that we believe that can be adapted, but for medical applications, I don't know.
That'd be an interesting idea, a genetic mouse where you can turn this on with doxycycline.
And it also depends that it can interfere with a lot of stuff. So it's something that needs to be considered.
Definitely. To end our webinar with one final question, the question you already teed up in your final slide. If one were to use a similar strategy to tackle other post-translational modifications, what would they need, or RNA modifications, if we take it one step further?
If it's other modifications, we can use in theory, we can use the anchors, can be the same anchors, but we can use different header domains for the modification, such as bromo domains or chromo domains to target other modifications. Another goal is, for example, if you look at 53BP1, that it also reads H4K20 methylation. We can also try to design a bivalent sensor as the one that we have in nature. So this has a two-door domain, so we can try to look at this. Think the anchor we can try to use, and there are now other anchors that don't bind the acidic patch, like bind other surface in the nucleosome. But with the same anchor, we can select different domains that will bind the post-translational modification, so ubiquitin-binding domains, bromo domains, for example, and then use a linker and try to target these other PTMs.
Great, wonderful. This was my final question. Thank you so much.
Thank you.
Renate, do you want to conclude?
Yeah, well, all right. Thank you so much for this interesting talk, Carolina, and for everyone for this lively discussion. If you have more questions, please feel free to visit our website, and you can still post questions there after the webinar or participate in the discussion in that Q&A section below the recording. And as I mentioned earlier, the webinar is being recorded and will also be posted later. So if you missed parts of it, you can go back to it in a couple of days or a week. And with that, we will end the session. Thank you again, everyone, for coming today, and I hope to see you again soon.
Thank you.
Thanks. That was great.
Thank you.
And I will see you guys in a bit.